Hello, everyone, and welcome to this webinar. Checkpoint molecules, Tregs, and induction of anti-tumor immunity. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by BioLegend. BioLegend creates world-class, cutting-edge antibodies and reagents across a diverse set of applications for immunology and cancer research. With over 30,000 trusted reagents and 100,000 citations, BioLegend is making an impact in labs across the world. Learn more at biolegend.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Dr. Vijay Kuchroo, Director, the Jean Lay Institute of Immunology and Inflammation, Brigham and Women's Hospital at Harvard Medical School. Dr. Kuchroo, you may now begin your presentation.
All right, thanks very much. Thanks for the opportunity to give this webinar. And I'm going to talk about checkpoint molecules. And they are expressed in multiple cell types. And we often think they only work on CD8 T cells. But I will give you an idea that how many cell types this checkpoint molecules can work on. I will also talk about these checkpoint molecules that are expressed on regulatory T cells and how they regulate anti-tumor immunity. And I often am rushed at the end, so let me describe people who really did the work.
I'm so sorry it looks like your screen is not sharing, so just one more time here.
So. Can you see it now?
Yes.
Looks great. Very good. Thank you. What I'm going to talk about is role of checkpoint molecules in different cell types. And we often think they work on CD8 T cells, but I'll give you some idea that others on impact of checkpoint molecules on other cell types. And the last section will be in the regulatory T cells. I want to acknowledge the work that was done by a number of people, and I'm often rushed at the end. So let me tell you upfront that most of the work I described today was done by Karen Dixon, Lloyd Board, Amin Sohail, Hugh Howe, Norio Chihara, and Asaf Mahdi in my lab. And a lot of work on T cell exhaustion was done by Anna Anderson's group, Nandini, Julia, Davide, and Lionel. They worked with me on this. Initial work on T cell exhaustion was done with Rafi Ahmed, and Dr. Jin was in his lab. And a lot of genomics work was done with Aviv Regev, Orit, Neil Yosef, Martin, and Elena at the Broad Institute. Now Aviv is at the Genetics Institute. Many of the mice and some of the data was generated in collaboration with Arlene Sharp. The crystal structure of TIM3 was solved by Rick Blumberg with Amit Gandhi, and many of the human data sets were generated with David Hafler and Tomo at the Yale University Medical School. And pathology was done with Ray Sobel. let me actually begin the talk. This is the outline of my talk. It's divided in 3 parts. The very first part is I'm going to talk about discovery of TIM3. And number 2 part will be a role of TIM3 not only in T cells, but it also works on DCs. And third part, which is new and novel and unpublished, is the role of another checkpoint molecule, TIGIT, on anti-tumor immunity. Let me go to the first part of the talk. A long time ago, in fact, 1986, Tim Mossman and Bob Kaufman first described Th1 and Th2 cells. Naive T cells, when they come in contact with antigen, they activate, expand, and differentiate. They acquire different effector phenotypes. And Tim Mossman and Bob Kaufman described that the Th1 cells produce interferon gamma, lymphotoxin, IL-2, and they are critical for clearing intracellular pathogens. But if you have a self-reactivity with Th1 cells, they will induce autoimmunity. And the Th2 cells are the ones that produce IL-4, IL-5, and IL-13, and their major role is to clear extracellular pathogens like parasites, and they are pathologically can induce the allergic reaction, asthma, atopy. And since Th1 cells were considered to be the cells inducing autoimmunity, we wanted to identify the molecules that are expressed on Th1 cells, and we immunized rats with Th1 cells, generated 20,000 different monoclonal antibodies, and screened them for over 6 months, and only to identify there were 4 different monoclonal antibodies that were selectively staining Th1 cells but not Th2 cells. And out of which the screen came a novel molecule, which we now call TIM3. And the antibodies to these TIM3, in fact, stain all interferon gamma producing cells. It also produces and stain the CD8 T cells produce interferon gamma, and they do not stain Th2 cells. We went ahead and together with Gordon Freeman, we cloned the gene. It was a new and novel gene, which has got about 302 residues long for mouse, 296 residues for human, has a signal peptide, IgV domain, a mucin domain, and a long tail, and tail has 6 tyrosines in it. And this is structurally, it has got the IgV domain, and this is the mucin domain, and the tail, which has got tyrosines in it. In fact, this was a founding member to identify a whole family of genes in both mouse and human. And in mouse, there are 8 members of TIM family of genes, and in humans, there are 3. And in humans, they are expressed in a chromosomal segment called 5q33, which has been repeatedly associated with allergy, atopy, asthma, and other immune-mediated diseases. In mouse, it's expressed on the centromeric end of chromosome 11, very close to where IL-4, IL-5, and IL-13 gene locus is. It has also been associated with taper, which is an allergic locus, and we now feel it's TIM3 that's regulating the function of many of the immune-mediated diseases, both in mouse and humans. So I'm going to talk about TIM3, and we initially wanted to identify where the ligand for TIM3 is expressed, and we made an Ig fusion protein of TIM3, and we screened a whole series of cell lines, and only to find out that TIM3 was ligand for TIM3 was highly expressed on CD8 T cells that have become tumorous, and the TK1 cell line is one of those. And many of the cell lines, tumor cell lines, in fact, will bind to TIM3 Ig, the soluble form of TIM3. We went ahead and used the TK1 cell line, the tumor cell line, and we identified this large protein mass, which was going from 40 to 60 kilodaltons. And when we deglycosylated it, and it turned out to be a very defined band of about 28 kilodaltons. And when we did a mass spec on it, and we identified the ligand to which TIM3 bound was galectin-9. In fact, this experiment was repeated 5 different times, and every time identifying a molecule, a part of TIM3, of galectin-9, which is the ligand for TIM3. So TIM3 really got a new lease of life since it is an inhibitory molecule. When the galectin-9 binds to TIM3 positive T cells, and these TIM3 positive T cells undergo cell death. And so when Anna Anderson was working in the lab, and we started doing a series of experiments, And one of those experiments is shown here, is that when you transplant the tumor, tumors progressively grow. They get infiltrated with these CD8 T cells. And if you analyze these CD8 T cells, that you will have CD8 T cells that don't express either TIM3 or PD1, the two checkpoint molecules. And some will express just PD1, and about vast majority will express both PD1 and TIM3 together. As you can see, that as they acquire these cell surface molecules, these are these inhibitory molecules, now called the checkpoint molecules. And as they acquire these checkpoint molecules, they progressively lose their effector functions. We in immunology initially used to call them co-inhibitory molecules, but they have been renamed, rechristened in cancer field as checkpoint molecules. So there is not just one checkpoint molecule. There is now a whole multitude of checkpoint molecules that have been identified, and their major job is to turn off T cell responses. T cell, when it comes in contact with antigen, it needs two different signals. As signal one is provided by T cell receptor specificity by cross-linked peptide MHC complexes, and the signal two is provided by a number of co-stimulatory molecules, and it puts the cell in cycle. When cell gets activated, it'll upregulate many of these inhibitory molecules, and they'll start producing IL-10. And the major job of these inhibitory molecules is to shrink the repertoire. So one of the initial experiments that was done is that when you transplant a tumor, a syngeneic tumor, on the mouse, and this was done in Anna Anderson's lab, and you can see that if you do isotope control and treat these mice with isotope control, the tumors will progressively grow. Sometimes anti-PD1 or anti-PD-L1 will work. Other times in this tumor, they will not work. This is B16F10 melanoma. But when you co-block both TIM3 with PD1, and you can see tumors literally melt away, and they also upregulate anti-tumor immunity. So here it is shown that when CD8 T cells infiltrate the tumor, they will upregulate these checkpoint molecules and TIM3 and PD1. Vast majority, about 50% of the cells will co-express these TIM3 and PD1 together. Some cells will express just the PD1, and others will not express any of these checkpoint molecules. And if you look at the effector functions of these cells, the cells that don't express any checkpoint molecules, they will make IL-2, TNF, and interferon gamma, shown by intracellular cytokine staining. When they upregulate PD1, some of the cytokines or effector functions are lost, but they are not completely lost. In fact, you can see in this slide that the PD1 positive cells started making more interferon gamma. But when you acquire both PD1 and TIM3 together, you can see majority of these effector functions within T cells are gone in tumor-infiltrating lymphocytes. What was also interesting, which Rafi Ahmed first described, is that we have these stem-like T cells that express a transcription factor called TCF7. And Rafi Ahmed first described that there's an antagonism between TIM3 expression. Once the TIM3 comes up, you lose this stem-like phenotype. And in fact, Anna Anderson independently discovered this as well, using transcriptional analysis, that upregulation of TIM3 results in the inhibition or loss of stem-like phenotype by loss of TCF7 expression. As shown here in detail is that that the stem-like T cells, which express TCF7, and their major role is to provide a reservoir of poised T cells and generate then effector T cells. And these are the cells where the PD-1 blockade works. And when they upregulate TIM3, and there's loss of TCF7, and cells become dysfunctional or exhausted. And so TIM3 and TCF7 are on the opposite poles of T cell function. And a number of elegant experiments done in Anna Anderson's lab have further confirmed it, and using CRISPR-Cas9 screens, and she is trying to figure out the biological mechanism by which this antagonism exists between TIM3 and TCF7 during T cell exhaustion. So having started this series of these experiments, and we wanted to ask a number of questions that are relevant to the tumor microenvironment and the development of anti-tumor immunity. And in this cartoon, what we are showing is that if we do single-cell RNA-seq analysis and now spatial transcriptomics, we can ask now a series of questions. We can ask that which cells express checkpoint molecules. And we often say that checkpoint molecules are expressed in CD8 T cells, but are there other cells that also express checkpoint molecules, PD-1, TIM3, LAG3, TIGIT, in tumor microenvironment? And what molecules in the tumor microenvironment upregulate the expression of these checkpoint molecules in tumor microenvironment? And what are the partners? Is it always this T cell is working on a tumor cells or other partners that we have not identified? And using the single-cell RNA-seq, we can begin to address these questions. And more importantly, when there's an interaction of the checkpoint molecules with these ligands on other cell types, what are the functional consequences of this impact? So using single-cell RNA-seq, we began to address many of these questions. And let me start with the first one. What are the molecules that are produced in tumor microenvironment that upregulate the expression of checkpoint molecules on immune cells? Early on, what we found out that IL-27, which is an immunosuppressive cytokine, comes during resolution of inflammation, was uniquely inducing TIM3 expression. And this is shown by qPCR, when we did a screen of almost all cytokines. And this is shown at the protein level. Indeed, when you activate Th1 cells or CD8 T cells, you give IL-27, vast majority of T cells will begin to express TIM3 on the cell surface. We were lucky enough that we had, in fact, started and begun an exercise where we were building these transcriptional networks for IL-27, and we will take naive T cells, we'll activate them with anti-CD3, anti-CD28, give them IL-27, and acquire RNA at 18 different time points. And using this, you can see that at the end of it, that you will have this TIM3 expression. These cells will start making IL-10, and we can build these chained networks. And it gives us a mechanism or understanding and building a hypothesis how IL-27 may be upregulating TIM3 expression in both CD4 and CD8 T cells. And safe to say that if you take wild-type CD4 or CD8 T cells or the IL-27 receptor knockout T cells, give them IL-27 following activation, there is a whole module of these molecules that can upregulate the wild-type T cells, which is lost in IL-27 receptor knockout mouse. And you can see that the TIM3 gets induced by IL-27, but that's not the only one. You can see a series of checkpoint molecules, TIGIT, LAG3, TIM3, and some of the CD39 family members, IL-10, all of them actually form a module. And IL-27 is not only inducing TIM3 expression, but in fact is inducing the whole lot of these checkpoint molecules. And the question is that how are they expressed, and are they expressed all on the same cell, and or IL-27 is acting on the cell, and different cells are expressing different checkpoint molecules. And doing a single-cell RNA-seq, we could indeed, in fact, see that the IL-27 will act on CD4 and CD8 T cells, and it will induce a series of transcription factors and induce the checkpoint molecules, including PD-1, TIM3, LAG3, TIGIT, and CTLA-4. And so they are all coming as a module, and it is not by a chance. In fact, the correlation coefficient of PD-1, TIM3, LAG3, and TIGIT is 10 to minus 23. And looking at our transcriptional network, we identified PRDM1 were the transcriptional regulators that IL-27 induces to upregulate whole checkpoint molecules, and including some of the novel molecules. So the paper was published, and this is the reference to it. And what we are showing is that normally IL-27 comes up during a resolution of inflammation, and it acts on effector T cells, and it induces these panel or module of checkpoint molecules. But tumors or tumor-associated fibroblasts and myeloid cells have co-opted this mechanism, and they begin to produce IL-27 to induce expression of all these checkpoint molecules on effector T cells that infiltrate the tumor. In mouse, we found that IL-27 is a key cytokine, but in humans, it's type 1 interferons together, independently or together with IL-27, that will induce these checkpoint molecules on the surface of effector T cells once they infiltrate the tumor. So just to conclude the first part of the talk, we identified TIM3 as a differentially expressed gene in Th1 cells and CD8 T cells. And it led to identification of the whole family of genes in both mouse and humans. I told you that it's in a unique place on the chromosome, which has been repeatedly associated with genetically linked to immune-mediated diseases. And I also showed you data that PD1 together with TIM3 will mark these terminally exhausted T cells, and anti-TIM3 and anti-PD1 together will induce anti-tumor immunity and tumor growth inhibition. And one of the interesting features of this study was that the checkpoint molecules, PD1, TIM3, LAG3, TIGIT, all come as a co-inhibitory module, and they get expressed together on the cell surface of these effector T cells. And I showed you some of the data saying that one of the suppressive cytokines, there may be others as well, that IL-27 will act on these cells to induce PRDM1 and CMF, and these are the transcription factors induced by IL-27 to upregulate and transactivate checkpoint molecules. Let me go to the second part of the talk, which is where are these which cell types are these checkpoint molecules expressed? And we often associate checkpoint molecules to be expressed on CD8 T cells. They clearly are expressed on those T cells, both CD4 and CD8 T cells and regulatory T cells. And in CD8 T cells, I showed you data that when they work together, they induce T cell dysfunction or T cell exhaustion. So the question is, are CD8 T cells the only cell types where checkpoint molecules are expressed? And so we, in fact, took an open-ended search, and Karen Dixon, who was working in the lab, and undertook a single-cell RNA-seq in animals that have been transplanted with the tumors, and took cells out of draining lymph node, non-draining lymph node, and from the tumor, and then manually annotated it. You can see these are B cells, these are DC2s, these are DC1s, and these are your tumor-infiltrating lymphocytes, and these are myeloid cells. Then she asked the question, why is TIM3 mostly expressed? And to our surprise, the TIM3 was largely expressed on the myeloid cells and DC1s. And this is your expression on T cells. And this is the protein expression. And what we did is we made a floxed allele and crossed them to a number of Cre drivers that will delete these TIM3 from T cells, DC, NK cells, and myeloid cells. And you can see that our flox allele is faithfully deleted. When you cross them in CD11C, you delete from DCs, but not from T cells. And when you cross them CD4 Cre, you delete it from from T cells, but not from the DCs. So it's faithful. And then we began to ask a series of questions that if you delete it with CD4 Cre, and TIM3 indeed is deleted on CD4 T cells, CD8 T cells, and Tregs. You transplant the tumor, and you can see some effect happens. There's little tumor growth inhibition, but not the massive growth inhibition that we had expected. If you delete it with the E8I Cre, which deletes in CD8 T cells, absolutely nothing happens. If you delete it in FOXP3 with ERT2 Cre and transplant the tumor, you can see the two curves superimpose each other, that it is not playing such an important role, at least in this tumor model, and this TIM3 is not playing such an important role on regulatory T cells in this tumor model. Then the question was, where does it have the most action? And the surprise came is that when we deleted TIM3 from DCs by using CD11C Cre, and you can see you transplant the tumor, the wild type floxed alleles have progressive tumor growth, when you delete TIM3 from DCs, you have tumor growth inhibition. But the CD11C CRE is not faithful to DCs, and we use more specific CRE drivers. And when you use ZBTB46 CRE, which is much more specific, and transplant the tumor, and you can see both MC38, MC38 OVA, in both cases, you have tumor growth inhibition. And the question is, what's the mechanism? So TIM3, which was discovered by expression on Th1 cells and CD8 T cells, and when you delete it, when you look at a single-cell RNA-seq, it's mostly expressed in the tumor microenvironment on DCs and myeloid cells, and deletion of TIM3 on T cells have little effect, but when you delete it from DCs using CD11C CRE and ZBTB46 CRE, you get this massive tumor growth inhibition, induction of anti-tumor immunity. And the mechanism looks like if you look at the single-cell RNA-seq data sets from the DCs from the wild type and knockout mouse, many of the components of the inflammasome pathway get activated in the knockout mouse. So what TIM3 is doing is acting in DCs, and it's also an inhibitory molecule on DCs, and it's keeping or inhibiting the activation of inflammasome pathway. And you can see here, if it's a combined score of enrichment in the tumor microenvironment, in the knockout DC, there's a massive activation of inflammasome. This data is published, and you can see that it's not on RNA-seq, but you can see also they both IL-18 and IL-1 beta, both are the components of inflammasome pathway, are activated in the knockout DCs compared to wild type, but there is no impact on the IL-12 P40 production from these two cells. So it looks like that the DC deletion of TIM3 results in activation of inflammasome pathway. Let me conclude the second part of the talk. In tumor microenvironment, TIM3 is predominantly expressed on DC and myeloid cells. There is some expression on T cells, and but dominantly is expressed on DCs. And when you delete it in DCs, there's an effective anti-tumor immunity, and this results in the generation of both effector CD8 T cells and these TCF7 positive stem-like CD8 T cells. And TIM3 suppresses DC function by acting and inhibiting inflammasome pathway. And if you neutralize IL-1 and IL-18 by giving anti-IL-1 and anti-IL-18 antibodies in the DC deleted mouse, you can abrogate the protective effect in anti-tumor immunity. So it looks like that one of the major mechanisms by which TIM3 operates on DC is to keep DCs quiescent and do not let them go and get activated to support inflammasome and produce cytokines like IL-1 and IL-18. So this was the second part of the talk, and let me quickly go the third and last part of talk, and I'm going to talk about another checkpoint molecule, which is TIGIT, and as you have seen that the TIGIT is part of this module of checkpoint molecules that comes up on exhausted T cells. So the question is what are the functional consequences when checkpoint molecules get expressed on T cells and it interacts with the ligands? What's the outcome of this interaction? And for this, I'm going to talk about this molecule TIGIT, which I told you is expressed together with other checkpoint molecules on T cells. And just to give you a brief introduction of what TIGIT pathway is, it's very similar to a CD28 CTLA4 pathway. On antigen-presenting cells, you have two ligands for CD28, B71 and B72, that interact with CD28 to give a positive signal. Cell goes into the cycle, gets activated, and upregulates CTLA4, and CTLA4 has a higher affinity for these two ligands, resulting in the inhibitory phenotype that you see in T cells, and CTLA4 will preferentially bind to these ligands and inhibit T cell activation. And TIGIT pathway is very similar to CD28 CTLA4 pathway, and TIGIT also has two different ligands, CD155, which is also called PVR, and CD112. And these two ligands will bind to the positive receptor, which is CD226, and give a positive signal to T cell, activate it, and then the TIGIT gets upregulated on activated T cells. It will steal these ligands from the antigen-presenting cells because of the ITIM motifs and ITSM motifs, and the TIGIT will inhibit T cell activation. So TIGIT initially was called in the genome as VSTEM3, and as I showed you that it has got an IgV domain, it has got transmembrane domain, cytoplasmic tail, and the tail has two ITIM motifs to suppress T cell responses. It comes up upon activation on all T cells, both CD4 and CD8 T cells, and we wanted to survey where is TIGIT expressed in tumor microenvironment. And when we delete TIGIT, and this is the global deletion using a global CRE, CMV CRE, and transplant the tumor, two different types of tumors, and you can see the TIGIT knockout deletion will inhibit tumor growth inhibition, similar to what we saw with the TIM3 deletion. So the question is, where is TIGIT expressed in tumors? And this is Lloyd Board, who did this experiment, is that he looked at both mouse tumors and some of the data he mined from the human tumors and looked at the expression of TIGIT, and this is single-cell RNA-seq. And if you look at these bar charts, is that in the mouse tumors, TIGIT is expressed a little bit on CD4 T cells, it's expressed on CD8 T cells, NK cells, and regulatory T cells. In case of humans, again, the story is the same, is that the TIGIT is expressed on CD4 T cells, CD8 T cells, NK cells, but there's a massive expression on FOXP3 positive regulatory T cells. So we generated a floxed allele of TIGIT, like what we did with TIM3. We crossed it to a number of CRE drivers, which were deleted in different cell types. And here it's showing that if you delete it by crossing this E8I CRE, you can delete it in CD8 T cells, but not from CD4 T cells. So our deletion and flox allele is faithful. Using these deleted mice, we begin to ask the question, where is TIGIT most important? And just to remind you that it is part of the checkpoint module, which comes on CD8 T cells. We have all assumed that its major role may be in CD8 T cells. So, but this is the test we did, is that if you delete TIGIT on myeloid cells by crossing it to lysine CRE, you can see there's no effect on tumor growth. If you delete it in dendritic cells, where TIM3 was most functional, absolutely nothing happened. And if you delete in B cells, there's a little bit of tumor growth inhibition. Then if you cross it to CD4 CRE, and you can see that there is a growth inhibition, so it must be working on CD4, because CD4 CRE deletes it on CD4 T cells, CD8 T cells, and also on regulatory T cells. So we thought it'll be all because of the effect on CD8 T cells. So we crossed the TIGIT flox allele to E8I CRE, deleted on CD8 T cells, and you can see that it had no impact on tumor growth. It's only when you delete TIGIT on FOXP3 FOXP3 T cells or the regulatory T cells, you get massive growth, tumor growth inhibition. And shown here is the same data in much individualistic fashion. You can see the deletion of TIGIT in Tregs induces tumor growth inhibition. And we can, in fact, see this growth inhibition in mice that have deleted FOXP3. In order to look at the ligand for FOXP3, for FOXP3 positive, TIGIT positive Tregs, And I told you there are two different ligands for TIGIT. One is CD155, which is a high affinity ligand. And we surveyed a number of tumor cell lines, and we all looked at whether TIGIT ligand CD155 is expressed. In fact, every tumor cell line that we have surveyed expresses CD155. This is true in both mouse and humans. And if you compare this to the myeloid cells on antigen-presenting cells, both CD11B positive cells and CD3 T cells also express CD155. And this is in the parenchymal cells, CD45 negative cells. The majority of CD155, the ligand for TIGIT, is expressed on these non-hematopoietic cells, and shown here, it's expressed on tumor cells. So it gave us an idea, maybe the TIGIT expressed on regulatory T cells may be directly interacting with CD155 expressing tumor cells. And here you can see is that we knocked it out, or we deleted PVR or CD155 from tumor cells. And this is the relative expression. You can see we have almost completely deleted the expression of CD155 on tumor cells. And we can transplant these tumor cells in vivo and take them out. And in fact, it's faithful that these tumor cells don't begin to re-express PVR or CD155 on tumor cells. So what I told you is in case of TIGIT, TIGIT is expressed on CD4 T cells, CD8 T cells, FOXP3 positive T cells, and in fact, FOXP3 positive Tregs and NK cells. And when we sequentially or selectively delete these TIGIT from different cell types, the major effect is when you delete TIGIT from regulatory T cells, the FOXP3 positive regulatory T cells. TIGIT has two ligands, and CD155 or PVR is the high affinity ligand, which is expressed on tumor cells. And we made a deletion mutant of CD155 in tumor cells, and gives us a way to identify the mechanism and the function of interaction of TIGIT on Tregs and CD155 on tumor cells, and in tumor microenvironment, how do they interact with each other. So we have now 4 different combinations where both the ligand and receptor is present. Ligand on tumor cells, receptor is on Tregs, and you can have the ligand on tumor cells, but receptor is missing on the regulatory T cells, or you have the receptor on regulatory T cells, but the ligand is missing, or both ligand and receptor is missing. And we transplanted these tumors into these FOXP3 positive, FOXP3 deleted, TIGIT deleted mice, and undertook a massive single cell RNA-seq, and then looked at what's happening in both the T cell side, on the regulatory T cell side, and what's happening onto the tumor cells itself. And you can see this is the pattern. You can see all the 4 combinations. And what was interesting is there was this selective expression of these a number of genes that's only present in tumor cells that have intact CD155, and they have intact TIGIT on FOXP3 positive T cells, which is missing when both of these receptor ligands are missing. And the question is, what are these genes? And it turns out that these genes, in fact, many of these are oncogenes, and the 4 of these are highlighted here, that when there's a receptor ligand interaction, the receptor on TIGIT on the regulatory T cells and CD155 on tumor cells, it looks like it's inducing or maintaining the expression of these 4 oncogenes, HAS1, PARP10, BRAF, and NRAS. All 4 of oncogenes are essential for the tumor growth and stem-like phenotype that you see in the melanoma of B16 F10 melanoma model. So what I told you is TIGIT is important for FOXP3 positive T cells in tumor microenvironment, and CD155 is expressed on tumor cells, and this interaction increases expression of these oncogenes in cancer cells. And this interaction we have also identified in using the Orion model, and thank you for collaborating with us. And we wanted to see whether we can visualize this interaction in the tumor human melanomas. And just to take you along, and we have these cells, these small cells with yellow rim. These are the CD8 T cells. The blue ones are the CD4 T cells. And these large cells with the staining and this S100 with cytox staining are all tumor cells. Just to take you a closer view is that you can see that the TIGIT is this blue color, and these are FOXP3 is purple color in the center. These are your FOXP3 positive regulatory T cells. This is CD155 rim on the tumor cells. You can see they are closely interacting with each other. And even in human tumors, melanomas, you can see this interaction of TIGIT positive, FOXP3 positive regulatory T cells with CD155 expressing tumor cells. So much of the data analysis suggests that this interaction is direct, and TIGIT expressing Tregs are interacting with tumor cells that express CD155, and they will induce the oncogenes. And one cell, they will upregulate and enhance the stability of FOXP3 positive T cells, so they become more immunosuppressive. And the other side, they are promoting tumor growth and tumor evasion by upregulating many of these oncogenes in tumor cells. Just to summarize what I told you in third part of the talk, TIGIT is part of the checkpoint module, expressing exhausted CD8 T cells. And in tumor microenvironment, we see it's also expressed in regulatory T cells. But it looks like the TIGIT is playing the most dominant role when it's deleted from FOXP3 positive regulatory T cells. CD155 is one of the ligands for TIGIT. It's expressed on tumor cells. And this interaction between TIGIT on regulatory T cells and CD155 on tumor cells increases stability and suppressive function of regulatory T cells, and it also reactivates or induces oncogenic program in tumor cells so that you make these stem-like tumor cells and that are capable of evading immune responses. And just to sum it up, what I told you is that we set out a series of questions, and first one was which cells express checkpoint molecules. And I gave you an ample evidence that it's not only CD8 T cells. We can also show you that they are also expressed on some of the myeloid cells and DCs like TIM3. And I showed you the data that IL-27 is one of the molecules, but not the only molecule that will induce expression of checkpoint molecules. And then I showed you some of these partners, these bedfellows we never thought will come together. And I showed you the data that, they act on CD8 T cells and antigen-presenting cells. Those are known players. But I also show you data that TIGIT expression on regulatory T cells will interact directly with the tumor cells in tumor microenvironment. It will reactivate oncogenes and make Tregs more suppressive. So that way, we can piece together the mechanistically how the tumor microenvironment is making an immunosuppressive environment where T cells get exhausted. Multiple different cell types will express checkpoint molecules, and they each have a different role in, like, gave example, TIM3 on DCs play a different dominant role, and TIGIT on Tregs plays a different role. Similarly, I think PD1 plays a very dominant role, and CTLA4 plays a dominant role in CD8 T cells. And this is my lab, and we have a soccer team called Kuchroo Lions. And if you have any questions later on, you can ask me live, or you can, in fact, write to me in any of these email addresses. I thank you for your attention. I'm happy to take any questions you may have.
Thank you for your interpretation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask Dr. Kuchroo, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question says, TIGIT antibodies have failed in the clinic. How do you reconcile the mouse tumor data with what has been observed in the clinic?
So the question is right, and in fact, there were a couple of clinical trials that were done, and this was done by Genentech and Roche, and they were trying to look at the lung tumors, non-small cell lung carcinoma, and there it has failed. But there are additional clinical trials that are still ongoing, and it will be interesting to see that what tumor type may be where TIGIT has got the most dominant role, and where the regulatory T cells may be playing an important role in their regulation. So clearly, the couple of clinical trials from Genentech and Roche had no effect, no efficacy, didn't show beneficial effect, at least did not meet the endpoint. But there are a series of these antibodies that were tested, and the antibody that Genentech and Roche tested had an Fc-active, which will not only delete and remove regulatory T cells, but will also remove the effector T cells. And this point was raised before, that whether there will be a difference in the efficacy of the antibodies, TIGIT antibodies, that are Fc active or Fc null. And there are a series of new antibodies that are Fc null that are in clinical trials. It'll be interesting to see what their results are and whether they will meet the endpoint. So let's wait and see what happens with those antibodies.
Thank you. Thank you. All right, our next question here. Our next question asks, did you look at the deletion of TIGIT in NK cells and its impact on tumor growth?
We didn't do it ourselves. And in fact, Mark Smith, when he was still active, had done a series of experiments where he deleted TIGIT from NK cells and saw no impact. So that's why we didn't do any TIGIT deletion on NK cells. But ever since that some of that work that was done by Mark Smith has become controversial, and many of those papers have been retracted. And we will need to actually go back ourselves to see whether TIGIT has an impact on NK cells. But at least in the data that was published by Mark Smith, there was no effect when TIGIT was deleted from NK cells.
All right, excellent. Moving on to our next question here. This question says, could you please share with the audience a brief comment of your opinion or recent discoveries you may have done about the effect LAG-3 both in CD4 positive and CD8 positive?
Which question number is this?
This is 9.
Question number, could you please share the audience a brief comment of your opinion on recent discovery you may have done with effect of LAG-3, both CD4 and CD8 cells? I apologize that we are in our lab are not actively studying LAG-3. It is done largely in the lab of Dario Vignali, and LAG-3 clinical trials, after a few hiccups, were finally successful, and they have in fact been approved for treatment with PD-1 in melanoma. I can only tell you what's published, and none of the work on LAG-3 has been done in my lab. And it does look like it's regulating the effector function of CD4 and CD8 T cells. And this is all published data from Dario Vignelli. Next.
Thank you. Our next question says, the part on TIM3 on DC for promotion of tumors. Did you dissect what types of DC, e.g. CDC1?
Yeah. So the TIM3 is largely expressed on CDC1 and migratory DCs. And so when you delete TIM3 from DCs, it's the MIG DCs and DC1s that expand and cross-present and induce CD8 effector T cell functions. So what you are raising is actually an important question, but that's not to say that it's not expressed on CDC2s, but the expression is much lower. But when you do deletion of TIM3, most of the effector functions are coming from migratory DCs and DC1s.
Wonderful. All right, our next question here says, is it known what ligand is important for TIM3 function in tumor DCs? Is it galactin-9? Could phosphatidylserine be important? I'm so sorry if I butchered that.
Yeah, so we exactly do not know which ligand is the most important ligand in tumor microenvironment. TIM3 has got this FG-CC prime loop, which forms this cleft in which phosphatidylserine from apoptotic bodies binds. So in DCs, we think it could be because there's a lot of cell death in the tumors, and the DCs, by interacting with TIM3, phosphatidylserine cleft, may be suppressing the DC function. But we have not formally proven it. So at this point in time, we can say that it could be either galactin-9 or TIM3, because the tumor cells do express galactin-9 after activation with interferon gamma. So we haven't formally proven, although our bias is that it could be the dead and dying tumor cells interacting with T cells and DCs through the CC prime FG loop, which binds to phosphatidylserine.
Okay, great. Our next question here that we have says, excellent talk, thank you. Do you think any other immune cells involved in this TIM3 or TIGIT medicated pro-tumoral function, like macrophages or neutrophils? Thank you very much.
For TIM3, we did do a myeloid deletion. We also did with lysine, we did same thing with TIGIT and myeloid deletion, and we did not see any impact on tumor growth inhibition when you delete from myeloid cells. But whether it plays a role in neutrophils, we do not know, because we never had a good access to a Cree driver that will delete TIM3 or TIGIT on neutrophils. So I do not know. We have no data on it.
Okay, great. All right, our next question here says, how do you see the duality of the checkpoint molecules in tumor immunity versus in autoimmunity? For example, T peripheral helper cells, or TPH, described in RA and MS express checkpoint molecules such as PD-1 and TIGIT.
Very good question. In fact, if you look at all the checkpoint molecules that have been discovered and are in the clinic and have been used in anti-tumor immunity, were discovered in autoimmune field, autoreactivity. And in fact, the checkpoint molecules set up a state of tolerance. There was a very interesting experiment that was done by Ken Smith at Oxford. He did an unbiased search trying to identify what are the molecules in human autoimmune diseases that may be linked to incidence of autoimmune disease, relapse rate, and response to therapy. And this was entirely unbiased. And in fact, he came up with this whole module of checkpoint molecules were essential for regulating autoimmunity. And they are setting the threshold for T cell activation, and they set up a state of self-tolerance. And when you delete them, these autoreactive T cells start getting activated. And this may be one of the reasons that the IAREs, that the side effects that you see with anti-PD-1 blockade and anti-CTLA-4 blockade, is the reactivation of these autoreactive T cells that induce tissue inflammation. So they are playing opposite roles. And in autoimmunity, they set a state of tolerance, and they have been co-opted by tumors. And in case of anti-tumor immunity, they suppress it. And that's why the checkpoint blockade works well enough in the patients to induce anti-tumor immunity.
Excellent, thank you. And we have time to just squeeze in one more question here. This last question asks, what is the role of checkpoint molecules in Treg functions?
Yeah. So the question is, what is the role of checkpoint molecules in Treg function? And it depends on the checkpoint molecule. The checkpoint molecule like CTLA-4, TIGIT, TIM3, all enhance Treg function. And they become better Tregs because of expression of checkpoint molecules. But PD-1 is an equal opportunity player. What it does in effector T cells, it enhances the effector T cell activation. And in case of Tregs, the PD-1 inhibits Treg function, and this is the data from Arlene Sharp's lab. So it is not blanket that they all enhance regulatory T cell function. PD-1 effectively antagonizes regulatory T cell function. But CTLA-4, TIGIT, and TIM3 enhance Treg function.
Wonderful, wonderful. All right, I want to thank you for your time today and your important research. We would also like to thank LabRoots and our sponsor, BioLegend, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye. Bye.
Thank you. Bye-bye.